And for once, I remembered to start it at the beginning of class. Okay, Lorraine, no problem. I saw your chat. Okay, so I'm going to go over just basic genetics and then I want to go through your homework. How did anybody struggle with the homework? I mean, you don't have to raise your hand if you don't want to, but yeah, it was a lot. It was a lot to go through. And so we'll go through the pedigree and the sex-linked traits, and I believe it had incomplete dominance on it as well. So share my screen here. Whoops, messed it up. Hold on. Okay. So hopefully you had a chance to read the textbook and go over the instructional videos. I think that they were pretty helpful. Okay, now I got my screen ready. How many of you had done genetics before, Punnett squares and things like that? Some of you. Oh, Chanel's saying Delsum for cough medicine. Yeah, I tried that and then I had Robitussin was the only thing that worked kind of. It tastes so bad. I was like, why does it taste so awful? And so I hated it. But yeah, I got like that cough medicine with codeine and it did not even touch it. So, all right. So I called it patterns of inheritance. I'm not going to go over Mendel's laws. I really want to get down to the Punnett squares and work on some genetics questions with you. Now, I think genetics is really interesting. Did I show you guys the epigenetics video? I don't think I did I? If we have time today, I'll show it to you. If not, I'll show it to you next week. So I gave you some terms to know, and we'll talk about these terms as I go through some of this stuff. And then, like I said, I have some genetics problems. I'm going to go to the whiteboard and I'm going to put some genetics problems in there for you guys to kind of work on, and then we'll go through them. So we talked about alleles when we went through mitosis and meiosis. So remember that an allele is an alternative version of a gene. Let me write that down for you. That's the technical definition. So write that down and I'll explain it a little more. So when I say the alternative version of a gene, so if you look at this image here, these are the same chromosome, right? These would be homologous chromosomes, meaning they code for the same traits, right? They have the same genes, but they are not identical because you could have different versions of the gene. You could have different alleles. And so here, the gene is for flower color. And the alleles are for purple flowers and white flowers. Now, remember when we're talking about genes, or I'm sorry, alleles, One allele typically is dominant and one is recessive. Now, when we talk about straight dominant recessive alleles, that's what we call Mendelian genetics. So Gregor Mendel, the father of genetics, as they call him, he determined that there were dominant and recessive alleles. But since then, we have found that there are a lot more variations like sex-linked traits and incomplete dominance and co-dominance. But that is what we call non-Mendelian genetics. So for here, standard genetics problems talk about dominant and recessive alleles. What is a dominant allele? Somebody tell me what the definition is, or give me an example.
The corresponding trait that the
Yeah,
allele is present in the genotype.
yes, exactly. The corresponding trait for like that allele, and it hides or covers up the recessive allele. Okay, so here the allele for purple flowers would be dominant over white. So we would give it, I don't know, I guess I would give it a P. I don't like using letters that look the same in upper and lower case because it's hard to tell what you're doing then. But for this example, we'll say that purple is dominant, right? Meaning that if there is a capital P in the genotype, the flower color is purple. So now I used a term that you guys need to know, which is genotype. So when I say genotype, I am referring to the actual alleles that a person has, right? So typically we have two alleles for a gene. And so the actual like letters are the genotype. So I'll write this. Possesses. That is not how you spell that. Possesses. I don't know. However you spell that. The alleles that an organism possesses for a trait or gene. Okay, so when we're talking about genotype, a person can be homozygous or heterozygous. So I'll wait for you to write down the definition of genotype and then we'll go over that. Okay, so when I talk about genotype, like I said, a person can be homozygous or heterozygous. So what does homozygous mean?
Same allele, like
Good,
A or B. Yeah.
good. The same allele for a trait, right? Homo means the same. So in my example, it would be big P, or little p. Now, like I said, you guys should have gone over this on your own. but I want to make sure that you have the correct information, plus repetition always helps, right? Okay, so an individual can also be heterozygous. Hetero means different. And so a heterozygous individual has two different alleles for a trait. Big P, little p. So remember when I'm saying big P, little p, big P, these are all genotypes, right? The actual alleles that a person possesses or individual organism. Hey, Brenda, are you able to stay after class for a few minutes? I think you
Um,
nodded. Yeah.
yes, I was hoping that that would have been a possibility.
Yes,
So yes,
absolutely.
I can stay. Thank
Excellent.
you. Yeah.
Okay, so we have genotype, but we also have phenotype. What is the phenotype?
The physical characteristic, like something
Exactly.
you're able to see.
Exactly. Good. And you can't always see it, but it is the physical, let's see, how do I want to say that? The physical,
Characteristics.
thank you, physical characteristic of a gene, right? And so I always think phenotype, physical. And so, like I said, you can't always see it because a lot of times genes code for, say, hormones or proteins. But usually, when I'm talking about genetics, I'll refer to physical appearance like eye color, hair color, things like that. Expression, that's the word I was looking for as well. Is that you that keeps answering, Alex? Or is that
Yeah.
Savannah? Good. I can see you've done
Sorry,
your.
I'm one of
Oh, no,
those.
no, no,
It's
no,
not
that's not what.
me. I'm not feeling too well.
Yeah, that's not what I was going to say, Alex. I was going to say I could tell you've done your homework, so excellent. And Savannah,
Yeah.
you sound about like I do.
Yeah, just about. I had to like medicate myself and
Yeah,
just call it a night
to-
early.
yeah, totally. I get that. Like half of you are sick, and so you all have my
Yeah,
sympathy. I appreciate you coming to class.
of course.
Okay. So when we're talking about dominant and recessive alleles, the only time a recessive phenotype shows up is if the individual is homozygous recessive. Okay, does that make sense to everyone? So in the case of flower color, the only time that the flowers are white is if they're little p, little p. Any questions on that? No. Okay. Can I erase this? All right. So let's jump over to the whiteboard. I hate the way this whiteboard works. It used to be different. Now I have to stop sharing, and then you guys are just like a little strip across the top of my whiteboard. Okay, so I am going to put the genetics problem here. And you guys don't really need to copy this down. I can post this worksheet after class. Make that bigger. Whoops, that didn't work. Okay, a purebred plant that produces yellow seeds is crossed with a purebred plant that produces green seeds. The seeds of all the offspring are yellow, why. So before you guys answer that, a purebred plant, what does that mean? Purebred. So purebred just means that the organism is homozygous. Okay. So when I say a purebred plant that produces yellow seeds is crossed with one that produces green seeds, why are the seeds all, the offspring all yellow? Who has an answer?
Because the yellow seeds are
Because
dominant.
Exactly. And you said yellow seeds are dominant, and that's good, Jess. And I would also say like the yellow allele, right? So if you look at it like this, I'm going to use Y here, big Y, big Y. So purebred plant that produces yellow seeds and a purebred plant that produces green seeds. So that would be like little y, little y. See, it's kind of hard to tell them apart when you use the um, letter that's the same in upper and lower. But all of the seeds, resulting seeds are yellow. And so we're going to look at this Punnett square. And this should make sense to you after the homework. I want to go through it anyway. I'm going to do my Punnett square. Okay, so when I do a Punnett square, I'm writing the alleles across the top and down the side. It doesn't matter whether you put yellow on top or yellow on the side, but you can't mix them up, right? I can't put an allele from yellow on the top and an allele from green on the top. So I put a big Y here, and then I attempt to put a big Y over here. And that did not work. Okay, so we have big Y here, and we have little y down here. Okay, so if I join these alleles in the squares, I have big Y, little y, right? Because I'm joining this allele here is coming down into the box, and this allele here is going into the box. Sometimes people get confused on how to make these individuals, right? Stupid thing. There we go. More or less, that's what it is. And so what this is representing is fertilization, right? So the, whoops, I wrote that wrong. That was supposed to be a big Y there. Sorry, guys. So, oh, it is a big Y. It's just not as big. Anyway, so these represent the alleles from this big Y plan, right? Remember meiosis, you have two chromosomes, homologous chromosomes, and during meiosis, you get gametes. And each gamete has one representation from the pair. And so that is what this is showing you. So this would be, say, an egg, and then down the side is a sperm. Remember, even plants have egg and sperm. So every box is a probability. This is not saying that there are 4 offspring. Okay, it's looking at the probability of getting a certain genotype and phenotype. So now all of the offspring, 100%, are heterozygous, and they're yellow, which means that yellow is dominant. Remember, the recessive phenotype is only present if the individual is homozygous recessive. Do you guys need me to write that down? I can only see 4 of you, so. All right, so let's go back. Actually, I'm going to do,
D.
D. I don't know if that was meant for us.
Sorry, I'm not sure if, like, the screen froze on my end, but I missed whatever you just said.
Oh, okay. Can you guys see my whiteboard?
I could see it, yeah.
Okay,
I just didn't
cool.
get the last thing that you said. Like
Okay.
it was like it looked like it was going off of like you were asking a question, but
Oh, no, what I was saying is that we know that yellow is the dominant allele because the only way that you can have the recessive allele is if your individual is homozygous recessive, right? And so if you have a big Y, your offspring is yellow, meaning that Y or yellow is the dominant allele, right? If it was white, all your offspring would be white. Okay, so let's do another problem that's a little more difficult. Okay, let's see here. How about this one? Oh, no. Too big. There we go. So achondroplasia is a form of dwarfism caused by a dominant allele. So a homozygous dominant genotype causes death. So individuals who have this condition, they're all heterozygotes. Okay. So we're going to use A as our letter for the allele. And it doesn't matter what you guys use for your letter. You can use anything, but you have to use the same letter for a trait. Okay, so a heterozygous individual is has achondroplasia, right? But they don't die. So if a person with achondroplasia mates with a person who does not have it, what percentage of their children would be expected to have it? So everyone go ahead and think about this question, work it out, and then we'll go over the answer. Oh, I just saw this present button. That's pretty cool. Oh, wait a second. Okay, so we know that achondroplasia is dominant. So we're going to give it, like I said, we're going to use A. So big A equals achondroplasia. And little a equals, we'll just say normal. Now remember, if a person is homozygous dominant, it causes death. Okay, so I'm going to go ahead. Oh, good. Nate, you gave me a 50%, big R, little r. Excellent. So we know that the male or just a person with achondroplasia mates with a person who does not have it. So we know that one person is a heterozygote. Otherwise, they'd be dead, to put it bluntly. So we have big A, little a. Now, I usually just put a times, like an X, right? Times little a, because the other person is normal. Savannah, good. So I see some answers in the chat. So let's do the Punnett square. Good, thank you, Lorraine. It looks like a lot of you have this down, so that's excellent. All right, so we have big A. I always write the dominant allele first. And that's just for consistency. It just keeps you from making errors. Then we have little a. Okay, so that's my first individual. Good, thank you, Savannah. And then I have little a and little a. Okay, so now I am just going to join up the alleles. So here we have big A, little a. Okay. We have little a. Another big A, little a. Whatever. Okay, that didn't work. There we go. And another little a, little a. So everyone who got 50% is correct. So it says, what percentage would be expected to have achondroplasia? So 2 out of the 4 squares have achondroplasia. So 2 out of 4 is 50%. Remember, you divide the numerator, I think is the top, right? Divide the denominator into the numerator. All right, does that make sense to everyone? Did I say that right? It's numerator on top, right? Okay. All right, so now what I want to do, it seems like everybody is doing really well with this. I would like to go over your homework. Okay. So your homework takes it a step further, right? First, we're going to do pedigrees. Now, I told you guys to skip. Oh, good, I took it out. Okay, so can everybody see this okay, or do I need to make it larger? You can always enlarge your screen. All right, so this is referring to people's ability to taste this chemical called PTC. And I have to say, I think I explained it up here. Oh, I did not explain it. That was on my other worksheet. So PTC, it's a harmless chemical, but it was discovered in the DuPont chemical factory. And I have to say that DuPont is, have you guys heard of DuPont? They make like cookware, they make all kinds of stuff, but they're actually not a very good company. They operate in, I think it's Pennsylvania. And so a lot of the chemicals that they use are those forever chemicals that stay in the soil and the water, and they're always in our bodies because of exposure to them. And they just do not break down. And so DuPont covered up the fact that their chemicals were causing cancer, a lot of these rare cancers in the community that they were based in, right? So a lot of people worked in the DuPont factory. It was kind of low income community. And kids were coming up with cancer and adults, and DuPont knew this because they had run studies on it and they covered it up for years and years. And so finally, Erin Brockovich, have you guys heard of her? Julia Roberts made a movie. She was Erin Brockovich. The real Erin Brockovich helped the community sue DuPont Chemical. However, people only got like, I think it was like $10,000 settlements, right? Their kids had died, they had cancer, and they get like $10,000, which to DuPont was nothing. And they still operate, which is unfortunate. But PTC was discovered in their factory. Some scientists were making whatever, and they got like this funny bitter taste in their mouth. And they were like, what is this? And so then they got a bunch of their friends and people to sample this chemical, which I'm sorry, somebody comes up to me and they're like, hey, you want to taste this chemical? We don't know what it is, and it gives a bitter taste. I don't think I would take part in that study, but I guess they found people who would. And so they figured out that there's one gene that codes for whether or not you can taste PTC. And so it's a dominant gene. And we know that because most people can taste it, right? If a gene is dominant, usually it's present in a lot of people. So you guys, for this pedigree, have to figure out the genotypes of all these people. Okay. So just to go over this, the basics, first of all, males are squares, females are circles. If it's shaded in, the person has that trait. And that applies to whether a trait is dominant or recessive. Now, what I do when I start a pedigree is I fill in all of the genotypes that I know. Okay, so if PTC is dominant, meaning shaded in individuals have it, that means if they're not shaded in, they cannot taste it. What does their genotype have to be if they cannot taste PTC?
Lowercase.
Exactly, homozygous recessive. So the first thing you do is you fill in all of these individuals that you know. So like John has to be little t, little t. And I'm not going to do the whole pedigree. I'm just going to do a little bit of it. Miriam, little t, right? Wendy, Anne, and Jean are all little t, little t, little t. Now, the fact that the offspring of John and Joan have little t, some of them, that means we can tell what Joan is. Okay. And so this is how you do this. First of all, when there is a line joining two individuals, it means they reproduced. And the line extending down represents their children. Okay, so everybody who is connected here, these are offspring. Now, Miriam is not an offspring. Miriam and Alan reproduced. So now Joan, we know that she's a taster, meaning she can be big T, or big T, little t. We don't know. Okay, she can be homozygous dominant or heterozygous. Now, I want to say here, you could not tell the genotype of all of these individuals. But now let's take a look here. Joan and John have offspring that are little t, little t. So remember, they had to get an allele from dad, so little t. And then they had to get an allele from mom, little t. So what is mom? Is she homozygous dominant or heterozygous?
Homozygous.
Exactly, heterozygous. The offspring had to have a little t from mom. And so we know that mom is heterozygous. But now let's look at Pauline and Keith. We know Pauline is big T, and then I usually put a line because I don't know what the other one is yet. We know dad, Keith, is little t or big T. We know their daughter, Elizabeth, is big T because they are all tasters. Now, Elizabeth reproduces with Tom. So what is Tom's genotype? He's a non-taster.
He's
Little t, little t.
Good, little t, little t. Now, Tom and Elizabeth have a child that is little t, little t. So that tells us that Elizabeth is heterozygote. I'm just going to put this over this. Oh, um, I thought it would let me click in there, but it won't. Okay, so we know Elizabeth is big T, little t. But now let's look at Pauline and Keith. One of them has to have a little t, but do we know which one? No. So we cannot tell Pauline and Keith's genotype with certainty. One is a heterozygote, but the other could be homozygous, dominant, or a heterozygote. We don't know which one. Okay, so Pauline and Keith, I think the question tells you to put a question mark. So it would be big T question mark, big T question mark. Does that make sense to everyone?
Just a little confused about is it Joan? Because
Joan.
they have kids with little t, so would hers not be big T, little t?
Oh, it is. It is. I had written this first,
Oh,
and
okay.
yeah, and I can't seem to erase these correctly. Oh, wait, is there an eraser? Oh, there is an eraser. There we go. Cool. So yeah, sorry about that, Chanel. Yeah, Joan is now, we know that she's big T, little t. There we go. Does that make more sense? Cool.
Yes.
Okay. So that is how you do this pedigree. And I believe there's another one for, okay, then there's another one for hemophilia. So we're going to go through that one. Now here, I wanted you to put the allele up here, and then you're going to join them in the box. As long as you did it some way that I can tell what you were thinking, that's fine. All right, so now let's look at sex-linked traits. Go to my whiteboard. Oh, but before I do that, actually, I was talking to Alex before a lot of you got here. I want to look at this particular disorder. Okay, so this is called, remind me what it's called.
Oh, Lynch's syndrome?
Yeah, Lynch's syndrome. Okay, and so this causes pigmentation, certain dark pigmentations on the body. And so this is inherited in an autosomal dominant manner. And so autosomal means not your sex chromosomes, right? You have 22 autosomal pairs of chromosomes. It codes for just your like body characteristics. And then you have your sex chromosomes, typically XX or XY. Now, this particular disorder is on, I believe I read chromosome 17, but like I said, it's dominant. So if you have the allele, you have the disorder. And so it would look like this. If I want to use L, mom here has big L, little l. Or she could be big O, actually. She could be either one. We will say mom is big O, big O. Okay, but dad has to be little l, because he does not have it. So mom could be homozygous dominant or heterozygous. So it's just like PTC. Now, you have two children that inherited big L from mom. Oh, mom is heterozygous. I'm sorry, because she has She passed on little L to two of her offspring. So let me fix that. Mom is big L, little L. Okay. So two of her offspring get big L, and two of the offspring get little l, little l. Now, the two offspring that are affected, what are their genotypes?
Big L, little L.
Exactly, they're heterozygotes. They had to get big L from mom, and then they get little l from dad. Now, the two children that are unaffected get little l from mom, little l from dad. Okay. Now, here's the thing. Here's what you were asking me about, Alex. Nobody in your family has this, right? And so,
Yeah.
what most likely happened is a random mutation on chromosome 17. It just arose with your daughter. Okay, so nobody in the family has it because it was like a random mutation that happened during meiosis or whenever for your daughter, right? So nobody had to have it. And in fact, only one chromosome had to have that mutation in order for her to get it because it's autosomal dominant. Does that make sense?
Yeah, a little bit more than what I was thinking.
Yeah, you were thinking more like sex chromosome, like X-linked trait,
Mhm.
but no, it's just a regular chromosome.
Thank you.
And actually, it's the NF1 that's chromosome 17. I'm not quite sure where this one is.
I couldn't tell
Okay.
you.
So now let's go to the whiteboard. And talk about sex-linked chromosomes. So sex-linked traits are on the X chromosome. They are not on the Y chromosome. The Y chromosome is really small. It has genes on it, but most of those genes are like there's the male determining gene, right? That codes for like more testosterone, things like that. And the X chromosome is pretty large and there's all kinds of genes on it. So an X-linked trait, like I said, is only on the X chromosome, or a sex-linked trait, same thing. Now, once or when as in a sex-linked trait, it can be dominant or recessive. And so your problem on your homework was looking at hemophilia, which is recessive. And so we're going to say big H equals normal. Little h equals hemophilia, which hemophilia is a problem with blood clotting, right? Your blood doesn't clot. Now, when we have a sex-linked trait, we write the allele as a superscript, meaning in the upper corner. And so I wanted you to use superscript on your homework. If you didn't, that's okay. So we write X. So let's say mom is a carrier, meaning she's heterozygote. She does not have hemophilia, but she carries that recessive allele. Now, I don't know if I can superscript this, so let's see. So mom would be X big H, X little h. She's XX because she's a female. So let's see here if I can superscript this. Does not look like I can. So what I'm going to do is make the X a little smaller. Okay, now dad is XY. He's a male. But let's say dad has hemophilia. So he is X little h. is super annoying. X little h y. Do that. Okay. I'm trying to make this so you can tell that they are superscript, more or less. Okay, so you do the Punnett square the same way as Mendelian genetics, dominant and recessive. Okay, so here's my Punnett. And now, you can stop me if you guys have questions as I go through this. So we're going to do X big H, right? And then we're going to do X little h. Those are mom's alleles after meiosis. Then for dad, we'll do X little h, Y. Now, I always put the X's first and then the Y. And I always put dominant allele first, just like I do with regular Mendelian genetics. Okay, so now I just join these in the different boxes. Okay, we have X big H, X little h in our first square. Oops. Okay, and same in this square. So what you can see here is that the daughters are 100% carriers. Now the sons will be X big H Y and X little h y. So 50% of the sons are normal and 50% are hemophiliac. Does that make sense to everyone? Now, so the homework asked you for probabilities, and if you put 25%, that's fine, but generally it's asking you, where am I? Out of the sons, what is the probability that they would have hemophilia? So let me show you what I mean here. Okay, so this is telling you the different genotypes, right? And so we know Prince Albert did not have hemophilia. So what is his genotype?
X, H, and then Y. I can't remember if
Right.
it's big H or not.
X big
X
H.
big H, right? He does not have hemophilia. So he is X big HY. Queen Victoria is a carrier. So she is X big H, X little h. And so when they reproduced, we got X big H, X big H, X big H, X little h, so a carrier. And then out of the sons, X big HY, X little hy. So here it says, what is the probability that they would have a hemophiliac son? It would be 50%, because you just look at the sons. You put 25%, I wouldn't count it wrong. Hemophiliac daughter, what is the percentage, or what is the probability that they would have a hemophiliac daughter?
50%.
Well, let's look at our daughters here. So hemophiliac would have to be X little h, X little h. So it'd be 0%. And Brenda, you asked me a question. Is it the same as Mongolian spots? Oh, the trait we were talking about, the autosomal dominant. I'm not sure. Alex, do you know if it's called that?
No, they're called cafe au lait spots or
Spots.
something like that.
Yeah.
But I'm not too sure. They might be like related in like the same group, just like different names, but I'm not
Mhm.
too sure either.
Yeah, I'm not sure either. Okay, so that is how you do sex-linked traits. Now, sex-linked traits are more common in males because they only have to have a dominant allele, and they have the disorder, whereas females have to have, I'm sorry, a recessive allele. Females have to have two recessive alleles. So then we did codominance and sickle cell. Now, this one is a little bit harder. Did everybody understand this incomplete dominance? Looks like I see a lot of yeses. So incomplete dominance, we use different letters for dominant or codominant, right? So we used a capital A and a capital S. There's not dominant recessive. They are both dominant. So big A is a normal individual, no sickle cell. Big A, big S, they have sickle cell trait, meaning some of their red blood cells are sickled, right? Because the big S codes for sickled and the big A codes for none. So you have like an in-between phenotype. And then big S, the individual has sickle cell anemia. Now, the interesting thing about being heterozygous, big A, big S, is that the person is immune to malaria. And I believe we talked about that before because malaria is vectored by a mosquito. It's caused by Plasmodium, which is like this little prokaryote that goes in, it enters your red blood cells where it reproduces. Then the red blood cell bursts and a bunch of little Plasmodium are released and they go on to infect more red blood cells. But they cannot infect red blood cells that are sickled. And so they don't cause full-blown malaria in people that are codominant, right? That are big A, big S. And so you had to do like regular punnets. And again, you're just writing in the traits and combining them in the punnet. And then you had to do this health profile. So you're looking at a family. So this is essentially just a pedigree, right? And you're looking at a lot of individuals. So the ones that are half shaded are big A, big S. The ones that are fully shaded are big S, big S. And then not shaded, they're big A, big A. And so you had to look at how many died of malaria, meaning they are what? What genotype makes you vulnerable to malaria? Who knows?
A normal, the
Mhm.
normal genotype.
Exactly. Big A makes you vulnerable to malaria. So all of these individuals had big A, big A. Now remember, we write it as a superscript. Individuals that are in good health would be big A, big S. And then 3 and 19 could have been big A, big S too, because they didn't die of malaria. And so for that, you would have to go down here and look at what they were. So 3 would be big A, big A. I think it was then 19 is big A, big S. Okay, and then what I wanted you to do, so you read through all this, you answered questions, you came down here. I wanted you guys to make a graph. You could have done it on paper, taken an image and inserted it into your homework, or done it on Google Sheets, whatever you wanted. So you needed to fill in this table. So family A is the one I gave you. You would just fill in the number of dead, add them all up, and then the percent of total deaths would be your total, or I'm sorry, the number of people with malaria divided by the total. So if you look up here, the number of people who died of malaria are, so we have 1, 2, 3, 4, 5, 6, 7, 8. How many individuals do we have total? We have 11, 12, 13, 16, 17, 18, 19, 20, 21. 38. Okay, and then I said that was 8. So it would have been died of malaria was 8. And the total of everybody in the table would have been 38. Oh, it didn't include good health. Oh, yeah, actually, that goes under. Okay, so you weren't supposed to include the good health people. So here, then your total would have been just dead people. So 1, 2, 3.
209.
Well, these are individuals. So it would have been 3, 6, 9, 11, 12, 13, 14, 18, right? 18 individuals. And so percentage of total deaths would have been 8 divided by 18, whatever that is. That gives you a decimal, and then you move the decimal place 2 places to the right, and that's your percentage. Or you multiply times 100, same thing. Now, neighboring communities, your total is 2,000. So total deaths would have been 1,080 divided by 2,000. Does that make sense to everyone? So what kind of graph should you have done? So you needed to plot a graph comparing the percentage of deaths from each cause. You had family A compared to the neighboring community.
A line
So,
graph or a bar graph?
good, a line graph or a bar graph. Usually a bar graph because that's what we use for categories. Causes of death would have been on your x-axis because that is the independent variable in a sense. And number of deaths would have been on your y-axis. And then you would have had 2 bars per cause, a bar for family A and a bar for family or neighboring community. And actually, it would have been percentage rather than number.
I did have a question for like before we get to the this table right there. You know, like the other question one where it was like list the numbers. You wanted like the actual numbers listed, right?
Yes, the actual people.
Okay, so it would have been like 2, 3, 7. Okay, I was
Exactly.
like, I wasn't too sure. It seemed too
Yeah,
easy for it to
I
be,
know.
so that's what made me question it.
Yeah. No, I wanted the actual numbers. Okay, so hopefully you all put a graph in there. And then this was just your comprehension check. Let me do this. Are there any questions on that? So I'm going to go through those. If you had like a lot of issues, I'll write comments on it and then you can meet with me if you have questions. Okay, so we're pretty much done. For Wednesday, we're going to do DNA structure and replication. Try to review it before Wednesday. I'm going to have you guys start on your homework that day as well. The Amoeba Sister video on DNA replication is really good. It goes over the, exact amount of information I want you to know. There's like a lot of enzymes involved in DNA replication, and I picked, I think, 4 that I want you to know. So. All right, any questions? Great. Well, I will see you guys on Wednesday, and Brenda, I will meet with you as soon as everybody logs off.
Thank you, Dr. Foltz. Have a great day.
Thanks. Thanks.
Thank
I
you.
hope you guys feel
Thank you,
better,
Dr. Foltz.
Savannah.
Have a good day.
Thank you.
Thanks. Brenda, I'm going to bring up my Canvas real quick.
Just really quick before you guys meet.
Sure.
Brenda had a question that she put down 15. Why was there 18? Brenda, are you asked? Because I was like, because I want to make sure I did
Oh.
that part right on my end. Because I was like, oh, it's kind of, I'm not quite sure if I did that correctly.